The Julius Kühn-Institut is the federal research institute for cultivated plants in Germany and part of the Federal Ministry of Food and Agriculture. Its activities focus on healthy and powerful plants in agriculture and horticulture, in forests and in urban space, as well as in cultivated areas as a whole. We work in the field of basic and applied research and on the transfer of research results into practice. The Institute of Ecological Chemistry, Plant Analysis and Stored Product Protection in Berlin studies the transport, fate and decomposition of agrochemicals in agroecosystems and their effects on organisms. It investigates the diversity of plant compounds and the protection of arable plants and plant stocks against pest infestation. Preventive and immediate methods of prevention, early detection and control are developed here. This is Thorsten Meiners, a scientist at the Julius Kühn-Institut with a parcel from Kenya. He and his working group are involved in the AflaZ project, which is funded by the Federal Office for Agriculture and Food. The package contains plant samples, which are to be analyzed and tested here at the Institute in the south of Berlin. The aim is to give recommendations to the Kenyan maize farmers about which naturally occurring plants they can use to control molds in maize cultivation. For this purpose, the plant samples are extracted to get their ingredients. The extracts are then analyzed. Volatile compounds are analyzed in a gas chromatograph with a coupled mass spectrometer. Here, the plant extracts are injected into the device and the compounds of different sizes are separated on a 25-meter column. In the mass spectrometer, the individual compounds are identified by their fragments. Non-volatile compounds can be examined in the high-performance liquid chromatography method and identified in the connected spectrometer. The effect of the plant extracts against the fungus can be tested directly. First, agar plates are inoculated with the fungus under sterile conditions. The fungal cultures are grown on corn agar in petri dishes in the incubator. The fungus is treated with the plant extracts and its growth is controlled. If the fungus continues to grow, the extract is not effective. If, however, the fungus stops growing or grows slowly, we have identified a plant that has an effect and can possibly be used to control black mold. Since the fungus produces aflatoxins, we also investigate to what extent the plant's active ingredients inhibit the formation of the fungal toxins. For this purpose, the treated maize grains are ground to make maize flour, which is then extracted. After that, the extract is analyzed by liquid chromatography and the concentration of fungal toxins is determined. In addition to direct control of the fungus, it is also possible to control insects which take the fungus with them by accident and thus spread it. The insect communities in cornfields in Africa are being studied together with the Julius Kühn-Institut of Crop and Soil Science in Brunswick and with the Kenyan scientists of KALRO, the Kenya Agricultural and Livestock Research Organization. At the Institute in Berlin, more than 50 insect storage pests are being bred. They can be used to test the effect of the plant extracts against fungi spreading insects. The extracts can be applied directly on the corn grains to see how the corn beetle Cytophilus siamaeus copes with them. Stored product protection scientist Benjamin Fürstenau is investigating the activity and survival of the beetles here. A locomotion compensator can show whether the plant extracts already deter the beetles with their odor. This means that if the beetle no longer runs towards the maize or maize flour as soon as the extract is added to the food, the plant has the potential to be used to combat beetle infestation and the transmission of fungal spores. After all tests are completed and the extracts analyzed, Thorsten Meiners discusses the results with his cooperation partners in Kenya. And once again, a package is going on a journey, this time to Africa. It contains the plant extracts that are now to be tested in the field to prevent the infestation of corn with Aspergillus.